Rogelio, you want to share your slides, and I will start introducing you. Okay, great. So thanks for coming to this week's Immune Zoom seminar. It's my great pleasure to introduce today's speaker, Dr. Rogelio Hernandez-Lopez, who is currently an assistant professor in the Department of Bioengineering and Genetics at Stanford. So Rogelio received his PhD in chemical physics at Harvard, and he worked in the Lestron Designer Lab on the mechanism of microtubule-based motors, kinesin and dyning. Then he moved to San Francisco and did a postdoc with Wendell Lim at UCSF. There, he attempted to address a critical question in cancer therapy, which is how to reduce the on-target but off-tumor side effect, because many of the tumor-associated antigens are also expressed in normal tissues, so at a lower level. So Rogelio designed, I think, a very elegant synthetic circuit that allows T cells to specifically recognize and kill cancer cells expressing high antigens, but did not touch the normal tissues. Actually, last week, Rogelio moved from San Francisco to Palo Alto. Actually, right now he is still communicating, but he's opened his new lab at Stanford. And at Stanford, I think Rogelio is combining his PhD training on biophysics and postdoc expertise on cell engineering, and aiming to answer mechanistic questions in cell biology and engineering multicellular system for novel therapeutic applications. Not surprisingly, Rogelio received numerous awards, including the prestigious postdoc fellowships from Cancer Research Institute and the UC Maxis. He also received Burrow Wellcome Fund Korea as a Scientific Interface Transition Award, and he is also a Chan Zuckerberg BioHub investigator. Beyond the research, Rogelio also actively advocates training in the next generation of students from all backgrounds. He is a co-founder and the director of the clubs de Ciencia Mexico and the Science Club International, which are nonprofit organizations that aim to expand the access to high-quality science education to youth in Mexico and Latin America. And in the past 6 years, they have served over 16,000 students through hands-on, in-person, and online workshops, which is really amazing. So without further ado, Rogelio, the floor is yours.
Thank you so much, Shaolei and Chuan, for the invitation and the introduction. I'm really excited to be here and share with you some of the work that I have done over the past few years. And as Shaolei mentioned, I just recently opened my lab at Stanford. But today I'm going to tell you the work that I did as a postdoc, and a little bit at the end, some of the future directions that I'm trying to take into my independent career. So just, briefly a summary, as also it was mentioned, I am trying to build a team and a research group that combines mechanistic cell biology studies with synthetic biology and cell engineering to design circuits that allow you to do bioengineering on multiple scales from proteins, cells, and tissues, and hopefully by using that approach, program and next generation cellular therapies for a range for applications. But one system that I would like to tell you today about is the one of T cells. I think in our minds over the past few years, T cells have been over and over, and they are actually a very amazing system to work with. Something that I'm like really fascinated about when working with T cells is that they perform like these very complex single cell and also collective behaviors. No, they can move around our bodies. They can perform like these very sophisticated identification tasks, like to decide whether something is a threat or is a part of our bodies. And in some cases, these T cells are also launching killing programs to kill foreign invaders such as virus or bacteria. So over the past few years, several groups and research around the world have been using methods of synthetic biology to actually engineer these T cells, to be able to kill cancer cells, and in particular, trying to redirect this cytotoxic activity of T cells against solid tumors. So the basic principle here is that you can use genome engineering and synthetic receptors, and in that way recognize particular proteins that are expressed on the surface of cancer cells, and redirect the cytotoxic activity of T cells against cancer. So this approach has been very, very successful in treating lymphoma, leukemia. These are blood cancers, and these therapies have been approved now in Europe and the US, maybe also in Asia, for the treatment of these diseases in adults and also in children. And there's a lot of excitement of trying to use a similar approach of this engineering of T cells to treat now solid tumors. The problem is that if you try to use the similar approach against solid tumors, there is multiple issues that need to be solved. And there is a few of them highlighted here. For example, when you want to use these T cells to track solid tumors, there is several problems such as finding what antigens you want to target on the solid tumors, how you control the activity of these T cells. You have to deal with the tumor microenvironment and also directing correctly these T cells to the site of where the tumor is, and allow the T cells to proliferate and divide and not exhaust. So the work that I'm going to tell you today has to do with this problem of the specific recognition of cancer cells and how you can control the activity of T cells to kill specifically solid tumors. And the problem that I'm gonna describe in the next few slides is the one of how you can train T cells to specifically kill target cells that have a large expression of a protein on their surface, because that's one of the main characteristics of tumor cells, but not too much or not do anything to cells that express the same antigen but at a lower level. So I'm going to describe something that in the field of I guess systems biology, synthetic biology is known as an ultrasensitive behavior or a sigmoidal behavior. That this will allow us to obtain this discrimination that we're looking for. So as I mentioned, the problem is that if you try to engineer these T cells against an antigen, for example, there is multiple proteins that have been studied for several years and are known to be overexpressed in tumors. For example, HER2, EGFR, GD2. These proteins are often in very large amounts in tumors, but they are also present at low levels in normal tissue. And if you try to make a CAR T cell against HER2, for example, what you will get is what is called a linear response, where regardless of the amount of the protein that is expressed on these targets, both of these targets are going to be killed. And in fact, I can show you some microscopy images of if you try to engineer these T cells against either cells that express low levels of HER2 or high levels of HER2, both types of target cells get killed. And this is a major problem because, for example, HER2 is expressed in your lungs and in your heart. And so there is the so-called on-target but off-tumor toxicity that in some cases has been actually being lethal. And ideally what you would like to do and what the field would like to do is to build, as I mentioned, sigmoidal response into these T cells, a behavior that is called ultrasensitive, where the only targets that are killed are those that express high levels of the antigen, but you spare the cells that express low levels. So the question that I try to address over the past few years is how we can control the activity of these T cells to discriminate targets based on antigen density. And as I mentioned, because of this response, the response that we're looking for is sigmoidal. We took inspiration from several strategies that have been used by biology over many years of evolution. No, so as you may imagine, there is multiple ways in which you can build this ultrasensitive behavior. For example, one very well-known case is the one of allosteric effects and how you can build. In the case, for example, of hemoglobin, like by multi-subunit complexes, you can have something that have this ultrasensitive behavior. But another strategy, and that was the one that we decided to pursue, is the one of building a circuit, a two-step circuit where you actually have positive feedback to build this ultrasensitive behavior. So I'm gonna explain in a moment the design of this circuit that I'm gonna use. And but before that, I need to introduce this new class of receptors that were developed a few years ago in the lab of Wendell Lim by Leonardo Morsut and Cole Roybal, both now professors, one at UCSF and the other at UC Santa Cruz. And so the idea here is that they took inspiration from the Notch receptor, that is a receptor involved in a lot of cell-cell contacts. And the idea was to keep the core of Notch, but replace the extracellular region of Notch for a recognition domain that you can use to target any signal of your choice. And what they also did was to replace the intracellular portion of this Notch receptor by an intracellular transcription factor, which is orthogonal to the cell that you are interested in engineering. And one feature of this Notch receptor is that upon binding of its ligand, there is going to be a cleavage of the intracellular region of the receptor, and then that should lead to transcription activation. So by keeping the core of this Notch into a new receptor that is now called syn-Notch or synthetic Notch, one can convert a binding event into a transcription activation event. So in those original papers, what they show is that you can actually use this synthetic Notch receptor in multiple ways. And for example, to sense contacts, induce differentiation, or multicellular patterning. And you can also use these receptors for therapeutic applications and for like more sophisticated programs, for example, to build Boolean or logic gates. But the way I use this synthetic Notch receptor is shown in this new slide, where what I did was to build a circuit where both the synthetic Notch receptor targets the antigen of your choice and control the expression of second receptor, in this case, the chimeric antigen receptor, that is gonna activate this T cell for killing. So in this case, both receptors are targeting the antigen of your choice, but there is a twist to this configuration, which is that the synthetic Notch receptor is of a low affinity for the antigen, while the chimeric antigen receptor has a high affinity for it. And the idea is that with these characteristics, the synthetic Notch receptor works as a high density filter. And then the chimeric antigen receptor should work as a output amplifier of this signal. And just to illustrate slightly better the way we think that this receptor should work and this configuration should work. So one should expect that if you have a T cell that expresses the synthetic Notch receptor that has a low affinity and encounters target cells that have a low levels of the antigen of your choice. So because the affinity of this receptor is low, the frequency of the interaction between the antigen and the receptor should be low, such as you shouldn't expect any CAR expression. But if the same receptor now this time is encountering target cells that have high levels of the antigen, the frequency of the interaction between the synthetic notch and the antigen should increase, leading to the transcription activation and expression of the CAR. And because the CAR is of a high affinity, then that should promote a complete engagement between the T cell and the target cell that ultimately is going to lead to the activation of the killing and also proliferation activity. So we thought that by using this design, we could actually build then something that has a sigmoidal response and be able to differentiate and discriminate target cells based on the antigen density. So in order to test this idea, what I did first was to build a series of target cells that the only difference among them is that they have different levels of expression of an antigen. And I took as a model antigen the case of HER2. So I made sure that the engineer lines that I built have levels that are in line and are representative of several cancer lines. For example, this is an ovarian line, SCOBY3 is an ovarian line, or BT474 is a breast cancer line that are characteristic of this overexpression of HER2. And I also had these engineered cell lines that have low expression levels that are of the same expression level that what you will find in, for example, normal tissues such as breast cells. So something that I want you to notice is that the goal in this case was to be able to differentiate 100-fold difference in expression levels. So something that is considered 3+ or overexpression of HER2 has in the order of 10 to the 7 molecules of HER2 per cell, while the normal tissues have in the order of 10 to the 4.7 or 10 to the 5 molecules per cell. And so these results and this analysis of expression levels is consistent with also a recent report of trying to map what are the level of expression of HER2 across a range of tissues. And something that like this group found is that indeed, if you compare cancer lines such as SCOBY3, that is ovarian cancer, these cells have like 100-fold more HER2 than a range of tissues that we have across the body. So we thought that being able to differentiate this 100-fold difference in expression level was a good test for this circuit that we're trying to build. So the other important piece for building the circuits that I described is having a series of recognition domains of multiple affinities. And in this case, we actually started with something that is a very, very high affinity. So the Herceptin antibody that has been studied for several years in the field of HER2 biology and cancers with HER2. So we started with that, and by introducing point mutations into the recognition domain of Herceptin, we were able to build a series of other scFvs that also expand roughly 100-fold difference in affinity. And these are some biolayer interferometry measurements to characterize the affinity of these different variants. So with these two tools in hand, we were able to build the circuits that we wanted and made assays to try to get at the question of whether by building this circuit, we could have a sigmoidal response. And what we found is that indeed, by using a two-step circuit, you can build new response, in this case, sigmoidal response into T cells. So in this case, what we did was to combine human primary cytotoxic CD8 T cells with the targets that have the different expression levels and measure after 3 days what is the leftover number of target cells into these wells, into these co-cultures. And we made the quantitation via flow cytometry. And we compare what we will get between using the T cells expressing the circuit strategy versus T cells that express constitutively these CAR constructs. No, and something that we notice is that, for example, in the case of the CAR, a constitutive expression of CAR, there is not a big difference between the high affinity variant with the low affinity variant, and these two variants actually don't have any discrimination. There might be some, but like it's minimal. No, when you compare a target that has high levels of expression level versus targets that have low levels of expression. So to characterize that this response, we try to fit the behavior of these curves to the well-known Hill equation that describes the ultrasensitivity of the system. And although this is an approximation, we try to put some numbers into the behaviors of these T cells and this response. So for example, by fitting these responses to this equation, you can get the threshold of activation, which in this case is roughly a 10 to the 5.5, and also the ultrasensitivity of this system, so which only is also in the order of 5. So in this case, we did a similar experiments, but across different donor T cells, and found very consistent results. So something that I would like to just emphasize is that there is two important pieces for the overall behavior of this strategy. So one, as I was alluding earlier, is the control of the CAR expression. And for example, you can label a CAR with a fluorescent protein in this case, and we use an M-cherry, and track what is the expression of the CAR as a function of the antigen density on the target cells that you are co-culturing with these T cells. And we found that actually the response of the CAR expression is linear as a function of antigen density. So you get much more CAR when you have the co-culture with targets that have high levels of HER2, and lower levels of CAR with targets that express low levels of HER2. But the important component here as well is the activation of T cell proliferation. So you can label these T cells with a dye, a membrane dye, and watch the dilution over time and over divisions of these T cells. So the larger number of the peaks here represent a larger number of divisions that these T cells underwent. And we found that there is robust activation of proliferation only in the case where you have the co-culture with the high density target cells. So in the cases of T cells that are co-cultured with targets with low levels, the levels of proliferations are similar to those that you will find with targets that do not have the antigen. So these two important elements are responsible for the overall behavior of sigmoidal response in the killing function. There is a question here, which antigen, affinity, or expression level? I think in the case of the T cell response, that's something that I think the circuit response is important to have this difference in affinity. I'll show you in a moment how you can, the affinity actually is very important. I didn't do much trying to vary the expression of the C notch, for example, but what I did is to try to vary the expression of the CAR as a constitutive vector. So you could have either a CAR expressed at high levels or a CAR expressed at low levels, and see whether, and those two could be of either high affinity or low affinity. But in none of those cases, like where you have a constitutive expression, you can find this ultrasensitive response or sigmoidal response. It's only the case where you have the circuit or the two steps where you can build the sigmoidal response. So I think in this case, the circuit strategy is a more robust way to build the overall behavior of a sigmoidal response. But yeah, maybe further studies are required to really pinpoint, like what are these important factors. So let me continue this, and then I'll go back to a few other questions in a moment, just to make sure that I go through the data that I want to show you, and then for sure I'll come back to some of these questions. So something that we wanted to explore further is whether we can actually tune the behavior. No, so you would imagine that for different types of cancer, you will need either something that is sharper in threshold, or maybe you want to have like a way to only target like the very, very, very high expressors, or maybe you want also to include some that have lower expression levels. So in a set of initial experiments, what we did is to fix the affinity of the synthetic notch, but modify the affinity of the CAR. And something that we found is that by lowering the affinity of the CAR, you can actually push a little bit the threshold of activation, but the consequence of that is also that you lower the overall activity of the system. And also you lose some of the ultrasensitive behavior that we were looking for. And also, not surprisingly, like if you increase this time the affinity of the synthetic notch, then you lose the overall sigmoidal response. And the reason for this is expected because then if you have now a synthetic notch that has a higher affinity, then you are not having this tight control on the CAR expression. So even when you are having target cells that express low levels of the antigen, you are going to start expressing the CAR. So you're going to get some killing of those cells. So, but at least from these experiments, what we found is that one can tune the overall behavior of this two-step circuit by playing with the affinities of these receptors that you use to build these circuits. So to further test this approach, in addition to these flow cytometry experiments that I alluded earlier, we conducted a series of microscopy experiments. So in the past, I showed you that if you have a constitutive CAR, either a high affinity or low affinity, both targets get killed. But in the case of the circuit strategy, the only targets that are getting killed are the targets that express the high levels of the antigen. So in this case, we use monolayers of these target cells that grow in 2D, and you use microscopy to track the depletion of these target cells. So this is the similar data, but now in some plots. And something that perhaps you notice from the videos is that while the circuit strategy is more discriminatory, it also takes some time to actually have the killing activity. No, and I guess further studies here are needed to really measure at what point the CAR turns on, and how this proliferation signal kicks in. But at least from this initial set of experiments, what is expected is that it will take some time for the circuit strategy to build, to activate the transcription activation, and then to build enough CAR molecules so that you can start the killing activity. But we showed that by this set of assays is that you have a highly discriminatory system in this case of the 2D monolayers. So the other set of experiments that one can do to really get at the question of whether there is going to be a specificity and enough localized activity of this strategy is to do experiments in mouse models. And for example, in this case, you can implant a tumor that expresses high levels of HER2 in one flank of the animal, and also another tumor in the contralateral flank, but in this case, that expresses low levels of the antigen, and inject IV these T cells several days after. And what we found is that the only tumor that is getting killed is the one that expresses the high levels of the antigen. So in the case of the a tumor that expresses the low levels, those tumors continue growing. And this is in remarked contrast, or actually this is consistent with to finding a larger amount of T cells in the high HER2 tumor, and also those T cells, if you pull them out and then use flow cytometry and look at the amounts of CAR that these T cells have, you will find a higher expression of the CAR in those T cells that are recovered from the high HER2 tumors. So in order to further test this approach, we use a few other types of tumor models. Something that you perhaps notice is that earlier I used those engineered cell lines that are very fast growing tumors. Within 20 days or so, the tumors could be such size that the animals have to be sucked. But we also use some other tumors that are grow slowly in these animals, and we show that the only tumors that are getting killed are the ones that express the high levels of the antigen. So this is a ovarian cancer or breast cancer, no, in this case, that express high levels of the tumor. And again, we were able to discriminate tumors that have almost 100-fold difference in expression levels. So the other type of experiments that you can do is to label the T cells with, for example, FLUC, and track the via bioluminescence the distribution of these T cells in the body of these animals. And we also found that there is a preferential localization of these T cells into the high HER2 tumors. So finally, in order to show that this strategy could be transferable to target other antigens, so what I did was to build a similar system, but in this case, targeting EGFR. So I found different variants of EGFR scFv antibodies and build CAR molecules and perform similar assays, no, with engineered target lines that have different levels of EGFR. And hopefully you can notice that regardless of the affinity of these variants, you don't get sigmoidal response. But in the case of having this two-step circuit where you use a low affinity synthetic notch to a high affinity CAR, but in this case against the EGFR antigen, then you can build this sigmoidal response to be able to discriminate targets that express high levels of the antigen versus targets that express low levels. So just to summarize this part of the talk, what I showed you is that by using synthetic biology and a very, very simple circuit strategy of synthetic notch, controlling the expression of a CAR, one can reshape the response of a T cell from a linear response into ultrasensitive response, and in this case, provide a new tool to discriminate tumor cells from normal tissue based on antigen density. And so hopefully this tool will be useful for targeting different types of solid cancers where you have like this type of a scenario. So there is other problems that I am now interested that have to deal with, for example, the tumor microenvironment, and that's something that I would like to briefly mention. And so one of my next goals is to try to use tools from tissue engineering and tumor organoids and bioprinting to try to get at the question of how the tumor structure affects the function of endogenous immune cells or these cell therapies. So I would like to get a quantitative understanding and also a mapping in how the structure of these tumors affect the activity of these T cells. So, so what I want to mention is that we started to get like some very preliminary results, but also in some like toy models of 3D structures to get at some of these questions. So for example, an initial set of experiments that we did was to build these 3D spheroids we use a cell line MCF10A that forms very easily spheroids. And as a first set of experiments, we had a line that in this case expresses high levels of HER2. So you can build these spheroids, embed them in Matrigel, have them in a matrix, put the T cells on top, and the T cells should traffic into these spheroids and either kill or not kill these spheroids. So for example, again, in the case of T cells that express constitutively a low affinity CAR, both types of spheroids get killed. But in the case of the circuit strategy, only the spheroids that have high levels of HER2 are getting killed. This was assessed via the amount of fluorescence that you will find of a caspase reporter into these spheroids. So this is the quantitation of that data that I just showed here. But the other type of questions that you can start assessing or answering is that of the spatial organization. So what happens if you have a tumor spheroid that has high levels of HER2, but is surrounded by other spheroids that have lower levels? So one question that some of the reviewers ask is, so what happens if you have this synthetic notch to CAR circuit and gets activated into a high HER2 tumor? Is that T cell gonna go now and move towards other locations and then start killing other tissues that you don't want to kill? And by doing this set of experiments of spheroids and we were able to start getting some data to answer this question. So what we found is that the killing is actually very localized. Only the spheroid that has the high levels of HER2 gets killed. And although there is some killing as a function of distance from that high HER2 spheroid, it only is restricted to maybe 100 microns or so. So beyond 100 microns, there is not much killing happening of those spheroids that are surrounding the high HER2 spheroids. But as you can imagine, more data is needed and other types of microscopy are required. So I've been getting my hands now into using light sheet microscopy and trying to get high spatial and temporal resolution to try to do this analysis. So for example, now a little bit over a year ago, I demoed a microscope to get at the question of what if we have like this high HER2 spheroid and then nearby we have a low HER2 spheroid. Are these T cells going to traffic to the other spheroid and start killing the low HER2 spheroids? But something that I found, at least in these experiments, is that the T cells remain very localized and the killing happens only into the spheroids that expresses the high levels of the antigen. So here, for example, again, these in cyan are the T cells, and this caspase signal is only localized into the spheroid that has the high levels of HER2. So one question that often have people have asked, and that's something that I'm very interested into digging more carefully, is the cases of heterogeneity. So what happens when in a single tumor you have different levels of expression, or you have like this so for example, there is cases of breast cancer where you will have regions or patches of high levels of HER2, but also low levels of HER2, or others where you have a kind of like a mosaic of cells that express high levels of one antigen and surrounded by cells that express intermediate or low levels. No, so the case of tumor heterogeneity is very relevant. So being able to sense different levels of antigen is going to be very important. Also, again, for example, in the case of PD-L1, which is often known to be immunosuppressor for these therapies. No, so it's known that the expression levels of these PD-L1 are could be very high in some types of tumors, but also the expression levels are variable. So I want to be able to design strategies to deal with different types of antigen densities. So by using these tumor spheroids of different compositions, different architectures. So it could be antigen density, or it could be different types of signals. One can start answering questions about how the tumor structure and composition affects things like the recognition, the filtration, and the killing. And by combining different strategies in the T cell side, engineering different levels of ultrasensitivity or strategies of recognition of these different signals, the idea is to try to modulate the overall behavior of these T cells. Now, as I mentioned, maybe for some types of cancer, you want to have actually a very broad response, and maybe for others, you want to have a very narrow response. Being able to tune the threshold of activation, that's something that's going to be very important. So I'm interested in modulating the activity of these T cells. So again, trying to get at the questions of how heterogeneous tumors affect things like motility and infiltration, cell-cell communication between the T cells, either engineered or endogenous from the immune system. So that is something that I am very interested to look more closely and to perhaps use these more advanced methods of microscopy and cell tracking to answer some important questions in cell biology and tumor biology. So the overall, I think, topic of some of the things that I want to do in the next few years are how one can use synthetic biology and, as I mentioned, methods of protein, cell, and tissue engineering to build communities of cells, either for therapeutic applications or to learn more about the biology of these systems. And here is just like a pictorial representation of some of the signals and some of the players that I would like to work with in the future. Of course, combining these tumor spheroids that have a defined spatial organization and control environment, you still need nice tumors of animal models of different diseases. So by combining this approach of engineering protein, cells, and tissues with animal models, hopefully one should be able to build a next generation of cell therapies for these solid tumors. And something that I'm very looking forward is to interface with different groups and different strategies. There is a lot of development in the fields of genome engineering and circuit dynamics, or single cell and spatial profiling technologies, organoid cultures, and 3D live imaging. So I think by combining like these different approaches, one can actually start getting into more sophisticated cell therapies for the treatment of solid tumors. So just to conclude, again, I just opened my lab. Here is my email address. If you're interested in maybe learning more about my research, or I'm actively looking for people to join my team. Here is my website and my Twitter handle. And with that, I'd just like to acknowledge Wendell Lim, my postdoc advisor, and my collaborators at UCSF and University of Helsinki, Seth Gardner, and the grad students that work with me, and technicians work with me, and of course the funding for my postdoc fellowship and for the project. And again, just I want to mention my lab is going to be part of bioengineering and genetics, and we're also part of the BioX initiative. I'm happy to answer any other question.
Okay, great. Thank you so much, Rogelio, for this wonderful talk. And I think all these are really exciting result, and also really, I think, very interesting future directions. And I'm sure like many people in the audience are interested in learning more in the near future. So we have a few questions. I think, let's see. So I see maybe try and raise the hand. Maybe try you go first, then we can come to the question in the chat box.
Okay, well, that's really exciting talk, and there's a lot of new ideas. I really like this circuit idea. But I have some really basic questions. So if I remember correctly, the low affinity to high affinity switch during the killing process, what's the kinetics for this switch? Like when you do the tumor cell co-culture or in vivo treatment, from the low affinity to high affinity, usually how long are you going to take?
Yeah, I think there is in the in vitro experiments, it usually takes out about in the order of hours to start the activation. So you want killing or you start seeing killing after 12, 20 hours. So that's why usually I have extended this assays in vitro for 3 days. No, where you can also see the effect of activation of proliferation and other programs. In vivo, usually the effects of like seeing whether a therapy is working or not will be in the order of days or weeks, not depending on what tumor model you use. Some tumor models are fast growing, others grow slower. So yeah, so in that case, to see the effect, it takes about a week or so or longer, not to actually assess whether the circuit or any therapy is working or not.
But that's just following this this point, because I saw the in situ tumor organoids you're doing with the microscope. I'm actually curious because if you say that in vivo, the switch of this high affinity to low affinity to high affinity take days or even weeks. I'm just curious why you think the T cells don't move around. I mean, they stick local and then still kill the high antigen expressed cells.
Yeah, that is a good question. My interpretation is that even if they move around, I mean, it depends on, and that's something that would be interesting to look like. What is the time scale of a complete biodistribution?
Right.
No, like
exactly.
How long does it take a T cell to actually explore the entire body? And I think something in vivo that also by bioluminescence, like the expansion is very localized. No, like you may find other signal. No, if you lower where do I see efflux signal? You may find it also in other places, but it's still like very diffuse. No, so the expansion, the localization, it's only happening in the tumor area, and something that I guess has to do with the way the circuit works is that it's not that you have the car and it will stay on, right? So there is also endocytosis of the car. So you need constantly the signal to replenish that pool of car molecules on the surface of the T cell. As soon as the high density or high the activation signal is reduced because you are killing those target cells, also you should expect to have lower and lower amounts of CAR on the surface of those T cells. So I guess that's another way in which this design helps with avoiding kind of like very far toxicity or far from the tumor area.
Okay, thank you. Yeah.
Okay, so let's see, in the chat box, I think Ian asked the question during the talk. So he or she has another question, which is, what if low affinity from off-target antigen present on normal cells, where it cause intensive off-target effect?
I am not sure that I.
So yeah, Ian, are you here? If you want to elaborate your question, I think what it means is probably it's worrying about the the off-target effect from the low affinity antigens on the on the normal tissues.
So with the circuit, if it encounters, and that's why that's what we set out to have, like this different 100-fold difference in expression. No? Like the affinity of the synthetic notch is such that if you put it in front of targets that have very, very low levels,
okay.
you shouldn't have CAR. There shouldn't be killing of those targets.
I see. Yeah, that makes sense. Okay, good. So Zhou-Xu, do you want to go ahead with your question?
Hey, Rogelio, really cool talk. I have a couple of questions. One, you mentioned this, Mr. earlier, that you mentioned CAR T have this linear dose response curve based on antigen densities. And how does the natural TCR behave? Do they have a linear dose response curves or natural switch-like response curves?
Actually, that's a very good point. The TCRs they have a sigmoidal response, and there is a and usually those TCRs some of them tend to have like a very high sensitivity, no? Like they can recognize there are very few number of molecules. And I think there it's because also you have the cooperative effect of the multi-subgenetic, no? Like you achieve that ultrasensitivity by building this multi-component complex. Given that, that's why, like now, not in the case of this avoiding the tumor toxicity, but in the case of, for example, in CD19, no, where you don't care about killing high expressors versus low expressors, because even if it's toxic, the toxicity is manageable. You actually care about killing cancer cells that have very low levels of expression. So now some of the strategies that people are using is trying to engineer TCRs because they know that have like a higher sensitivity and get at things that will kill even targets that have very low levels. But that's a good point. I think to build this type of strategies, one can use other or this type of behavior. So one can use other strategies such as building this multi-component, no? Or for example, you can think about multivalency, like having low affinity or multivalent interactions that should also have this type of sigmoidal response. You could also use things like inhibited receptors, that some sort of get uninhibited by the density of the target. So yeah, so I think one interesting direction could be also trying to get that, whether some strategies are maybe faster than others and maybe it's more sensitive than others, how you layer those different strategies, such as you can modulate the overall behavior of these T cells.
And since you mentioned ultrasensitivity, my follow-up question is, there are different ways you can achieve ultrasensitivity. You mentioned two, right? You can overlay different circuitry, you can have cooperation, or you can have positive feedbacks. In these cases, I think the T cell proliferation give you a natural positive feedback, which is very surprising to see. With CAR T, you actually see a linear curve. Do you see a decoupling between the killing threshold versus T cell proliferation threshold in the CAR T cells?
That's a good point. And I think that's something that I haven't explored that much, but I always thought, you cannot do both, right? It's either kill or to divide, no? And there should be some sort of decoupling or a way in which the cells as a community interpret either some of you are going to be killing and maybe others are going to be dividing, no? And there is the entire other layer of once the population sees antigen, like some of those cells are going to start differentiating into central memory or like stem cell like. So I think there is like other layers which each of those subpopulations has different rates of proliferation. So there is still few other questions that I think are going to be important to really know how you control the proliferation and which subpopulation is the one that is going to be important to control or modify. So I think that's another way to look at it.
So my last question, kind of related to maybe Ian's question a little bit too. Have you guys tried to just mix the high density cells with the low density cells and then try to kill it to see
Yeah, that's
if you have
a
quite
good question
damage.
as well. I have done a little bit of that, and that's something that I also want to explore further. So something that I found, just I don't have the data here, I think, depending on the ratio of those populations, either you are gonna get only killing of the high HER2 or the killing of everything. So for example, if in a single well you have the majority of them high HER2 with few of them low HER2, you wipe out the entire population. But if you have now the majority being low HER2 with few that are high HER2, then it is very specific to the high HER2. And this comes back to the point, no, that if you eliminate the priming signal, no, or this activation signal, then there is no CAR anymore. No, so it's only the time that the T cell still has the CAR that can go and do off-target killing, but it's not, it doesn't spread. No.
Cool, thank you.
Okay, so next, I think Chenji raised his hand first. So Chenji, do you want to go ahead with asking a question?
Thanks, Charlie. So it's a wonderful talk. This is Chen Qixu from Shanghai Institute of Biochemistry and Cell Biology. It's really fascinating to hear the toxicity strategy. So I'm just thinking, so this new type of CAR strategy, have you studied the behavior of these CAR T cells, in particular in vivo? So I'm thinking about the persistence feature, like in the memory population. When you compare with conventional strategy, do you see a similar behavior or different, like a metabolic program and other stuff? Just curious how this cell behave in vivo. Like, can this cell last a long time?
That's a very good question, and that's something that me and others in the LIM lab have started to explore further. No, something that we are finding out, and I think is consistent with what other groups are finding, is that not having the CAR expressed all the time, it's actually very beneficial to the health of the T cells. No, like or rather, no, like having the CAR expressed at all times is inducing exhaustion, is like less all these markers, no, smaller fraction of stem cell-like T cells. Those are the ones that you want to have for the therapeutic efficacy. So yeah, we have been trying to, not metabolic, but at least profile like some of these markers of exhaustion, like the fraction, and we found that the synthetic notch to CAR strategy is very good at keeping the health of these T cells in a more naive stem cell-like state. A few other groups are finding, no, that if using these destabilizers of CARs or things that keep the CAR off, that also promote this type of kind of like reinvigoration of these T cells, because there is no signaling. No, like it seems that just having the CAR construct there expressed on the surface of these T cells is already generating some type of signaling that is detrimental to the overall health of these T cells. So yeah, I think there is still more research to be done to, again, not to get at the question of what aspect of this control is more important. No, is it the metabolic profile? Is it the transcription factors that keep the different populations of effector and naive and stem cell-like? No, but yeah, I think that's a very good point.
Okay, thank you. So a follow-up question: how long this CAR molecule can be sustained on the surface?
Yeah, the off rate, we have tried to measure it. I think it's in the order also of hours, at least in vitro. I don't.
It's quite dynamic without.
Yeah.
Without this, okay. Well, so another, it's an unrelated question. It's think about the antigen loss that's often observed in blood cancer treatment. So like you mentioned, in this CD19-based CAR T therapy, toxicity is not really an issue, but antigen loss will be a big issue at later time point for relapse. So how do you think about, how do you design a different strategy to deal with this issue?
Yeah, that's a great question. I think that at least I'm not saying that it's going to be for everything, but in the case of HER2, apparently, at least from my conversations with clinicians, they think that antigen loss is not going to be a big issue. Not big, like apparently, like the amount of HER2 that is in overexpressed tumor lines or tumors, it's so massive that they don't think that that's going to be an issue. But I think if that becomes a problem, then that's where you need to bring these other strategies of either using like multivalent CARs or maybe strategies that allow you to target other antigens or to have like this widespread activity but localized, so that you also kill cells that do not have the antigen. And I think it's more and more now known that there is these effects, where just by secreting, like these different chemokines, cytokines, and interferon gamma, TNF, that the T cells are expressing once they are activated. That might also be toxic for the cells that are around that are not expressing the antigen. But yeah, that's something to keep in mind if that problem arises.
Okay, thank you so much.
Okay, so our next question came from Liu Baichuan. So Baichuan, do you want to ask your question? Or I can just read the question in the chat box. Do you think the current poor therapeutic effect for solid tumors is due to the non-optimal target selection or the inhibitory effect from tumor microenvironment? As Ewing's sarcoma seems to respond well to CD99 CAR therapy.
Yeah, this is a very good point. I think there is, as I mentioned, there is multiple problems now, and it seems that in some cases is, as you mentioned, like which is the correct or what's the appropriate antigen to target. In other cases is how you deal with the microenvironment, like all these signals that the tumor are presenting that generate like less infiltration, like the entire thing of like hot and cold tumors, and the immunomodulatory either secreted or presented on the surface of these tumor cells or the other cells that are in the environment that are inhibiting the activity of the T cells. I think there is still a lot of questions that need to be answered to really pinpoint what is the most important aspect of the tumor that prevents these T cells to have their full potential. Yeah, I think that's something important to consider.
Okay, good. So if possible, I'd also like to ask like two questions. So first, I just wonder, so now you have the low affinity Sigmoidal and the high affinity CAR. What will you expect if you switch these two? If you have a high affinity Sigmoidal and a low affinity CAR, will you still see a higher heal coefficient?
I think I have something that is not exactly that, but the intermediate that I think I should. Oh, let me see. You lose the. Yeah, that's something that probably I need to go back and look more carefully. But if you increase the efficiency, the affinity of the synthetic notch, you lose the ultrasensitivity. At least in how this is calculated or estimated. It seems that now the heal coefficient is lower than before.
Yeah, I think it's interesting to see if you combine the two. Yes, because if you just manipulate one, it's kind of predictable. But I could see the problem of that one because you don't want to have always a high expression CAR, which might cause some exhaustion problem. But just curious from the theoretical perspective. If there's
Yeah,
any-
something for example that I am ignoring here and that I've been finding there is actually an optimal affinity for the CAR, and actually having a high affinity CAR might not be the best choice. And the reason for that is that you also have this problem of trogocytosis and self-killing. No, so if the CAR is of a high affinity, then you are also getting more antigen into these T cells. These T cells start killing each other, and that's something that you kind of ameliorate by lowering the affinity of the CAR. So even with this circuit strategy, you still have that problem. So I guess the short answer is there is an optimal affinity also on the CAR that having the highest might not be the best choice.
Yes, yeah, that's also a good point. So the other question is there any mouse model to evaluate this on-target off-tumor toxic effect? Let's say, is there any model that we can evaluate, for example, the damage to the liver, to other
Yeah,
tissues?
that's a very good question, and that's something that I don't think that there is few models that have been proposed, and but then all of them have caveats. No, for example, using AAV, I think it's a few years ago, Carl June has this model where they can deliver HER2 to the liver, no? So they can express at low levels, and so you can compare what happens if you implant a tumor sub-Q versus the liver. So that's one case, but still, I mean, that's how much you put in the AV and like whether those levels are comparable. No, the other way to perhaps try to do it is going to a syngeneic mouse model and trying to target a mouse antigen. No, but then you have to deal with optimizing the affinity of these scFvs, no, so that you actually have good targeting of the antigen that you want. No, yeah. So and then there you are studying. I guess you are studying there like the discrimination, but like how much whatever you find there is going to translate to the human case. That's another question.
Yes, hopefully will be some good new model developed in the future. Okay, so we have another question coming from the chat box from Daisy Liu. So Daisy, I wonder if you want to ask question by yourself, or I can read out. So is there a defined KD range for the combination of high and low affinity antibody for high threshold targeting in all tumor, or it changed in other tumors?
That's a good question as well. I mean, like for example, in this case, the difference in affinity between the low and the high is 100, I think, if I remember correctly. But that doesn't. I guess that doesn't necessarily mean that that's gonna be the always gonna be the case for other types of cancer. So it's gonna be dependent on what the antigen expression level of the different one, and whether these scFvs are targeting, what are they targeting? Are they targeting what epitopes? Like it seems that there is some difference in activity also, whether they target regions that are further up from the membrane versus closer to the membrane. I think there is a lot of things that still are to be understood in terms of how these molecules work. It's actually amazing that they work not just by putting these receptors they kind of like use redirect the clean activity of this. But I think yeah, at least in this case, that seems to be the case.
Yeah, and also I think like the cell adhesion molecules also play an important role here, especially for the low affinity ones. So I think ideally, if there is I think integrated parameter can predict, let's say, okay, what will be the optimal KD for a specific tumor antigen. I think that will be
yeah. Well, yeah, and that is I totally agree with you. And then the other thing that I probably at this moment I'm using as a black box is that there is this step of the transcription activation, no? And there is like binding domains there, the affinity between the transcription factor and the binding and the number of sites. That is also another layer, no, that you could tune, regulate to change the behavior of this.
Yes. Okay, so that's great. I think that this is a very nice discussion. If there are no more questions, let's thank Rogelio again for this wonderful talk. So yeah, hopefully you will enjoy the starting of your lab period. It will probably not be very relaxing, but I'm sure it will not be boring, and you will find many surprises every day, whether it's good or
yeah.
Okay.
I'm looking forward to it. Yeah.
Cool. Thank you. Yeah. Thanks, Rogelio.
Thank you. Thanks again for the invitation. Yeah. Thanks everybody.
Absolutely. Okay. See ya.
See ya. Bye.